Hi, good morning and good afternoon, everyone. Welcome to today's webinar, A Workflow for Knock-in Genome Editing, Simplified. Our presenter today is Dr. Matthew Poling, Product Manager for Genome Editing at Thermo Fisher Scientific. My name is Louise Baskin, also of Thermo Fisher Scientific, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Invitrogen by Thermo Fisher. Invitrogen offers tools and solutions for every step in the CRISPR genome editing workflow. For more information, please visit www.thermofisher.com/CRISPR. Okay, let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you like at any time you want during the presentation. To do this, you just type them into the Ask a Question box and then click on the Send button. A portion of the questions will be answered live by Dr. Poling following the presentation, and all questions that don't get answered live will receive a written response by email. You'll also see multiple tabs at the top of the page. Feel free to click through those. They have related applications, upcoming and related webinars, as well as links to some different resources on the topics that you'll be hearing about today. Also, please notice that you can share this webinar on your personal social media. Just click on the social sharing tab to let your friends and colleagues know about today's live event. If you have any trouble seeing or hearing the presentation, click on the support tab that's found at the top right of the presentation window, or you can report the problem by clicking the Ask a Question box. This presentation is educational and offers continuing education credits. So click on the Continuing Education Credits tab located at the top right of the presentation window and then follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Matthew Poling. Matt earned his Ph.D. in Biomedical Sciences from UC San Diego and joined Thermo Fisher Scientific in 2015. He's currently the lead product manager for genome editing technologies and works very closely with Thermo Fisher's research and development scientists to create innovative genome editing tools and services. So that is all I have to say. Matt, I'll go ahead and turn it over to you for the webinar.
Thank you for that introduction. I will go ahead and get started with today's webinar. Before we get too deep, I did want to take a moment and acknowledge our genome editing R&D and product development group that I work very closely with. We're all located in Carlsbad, California, and there's a team of 25 full-time genome editing scientists that I partner with. We're all working to advance tools and application development around genome editing, working to improve the CRISPR-Cas9 and TALEN effector nuclease technologies, and with specific emphasis on functional genomic applications. Our Carlsbad site acts as our R&D hub and a center of excellence. We have a number of different R&D teams there, including our delivery and transfection groups, our stem cell biology team, and are able to utilize resources from our NGS platform, as well as our antibody development and custom services teams. And the R&D team here has done really a great amount of work here, and I'm going to be displaying a good amount of their data today. For today's presentation, our agenda will consist of a review of knock-in genome editing. We will then present the rules for successful HDR donor design. We will demonstrate how to easily design your own knock-in tools with our TrueDesign Genome Editor. We will then show how to obtain 100% edited cells with TrueTag donors. And then lastly, go over 2 case studies that apply TrueTag knock-in editing for pluripotent stem cells and T cells. So to have a quick review of genome editing, what we're trying to accomplish here is looking at double-stranded DNA that's in the genome of the cell line you're interested in, and using an engineered nuclease to cause a double-stranded break in that DNA. After that double-stranded break has occurred, we're going to use the cell's natural repair machinery to repair that double-stranded break and cause the genome to be modified in a way that's relevant for your application type. If you're looking to do gene knockouts, this is done by non-homologous end joining, which will cause indels, insertions and deletions, that can cause frame shift and premature stop codons in your gene of interest. This is done to produce knockout cell models as well as do genomic screenings. The second pathway is to do gene insertions or knock-ins by HDR. And this is a little bit different than what's done for gene knockouts, and that includes a donor molecule with left and right homology arms flanking your insertion sequence that would get incorporated into the genome. These kind of knock-ins are used to add reporters to genes, create fusion proteins, disrupt functional domains, or change SNPs to mimic disease models. And this knock-in approach is going to be the main focus of our presentation today. For a knock-in edit, there are a number of required materials that are needed. First, a specific and efficient nuclease system. Second, a repair template with the desired change that you want to have incorporated into the genome. And third, a method to deliver these materials into your cells. And for today's presentation, we're going to focus on number 1 and number 2 specifically. We'll talk about different types of specific and efficient nuclease systems, as well as our recommendations for the repair template and how to incorporate that desired change. The two main nuclease systems that we're developing and working with at Thermo Fisher is the CRISPR-Cas9 system, which I imagine a large majority of our audience members are already familiar with. Briefly, CRISPR-Cas9 system is a bacterial adaptive immune response pathway that consists of the Cas9 protein, as well as a duplex of CRISPR RNA and tracer RNA molecules. The specificity of the cleavage by Cas9 is dictated by the CRISPR RNA sequence, this 20 nucleotide sequence that can be designed to any part of the genome, assuming that it's next to a protospacer adjacent motif or a PAM site, which is required to be next to that CRISPR RNA sequence. For Streptococcus pyogenes Cas9, which is the most predominant Cas9 we see scientists using, that PAM site is NGG. When you design your guide RNA to be specific to your target genomic loci and is adjacent to a PAM site, you'll get cleavage 3 bases upstream of your PAM site, and this will produce blunt-ended DNA breaks. And Cas9 and these guide RNA molecules are introduced into eukaryotic cells in either a plasmid format, as RNA molecules, using a Cas9 mRNA and your guide RNA molecules, or as an RNP or ribonucleoprotein format, in which a recombinant Cas9 protein is complexed with a guide RNA in vitro, and then that protein guide RNA complex is delivered into your cells. The second nuclease system that we work with are TAL activator-like effector nucleases, or TALENs. And TAL-based genome editing uses these TAL effector proteins, which have been engineered from bacteria to be specific DNA-binding proteins with functional domains fused onto them. The TAL effector proteins consist of repeat amino acid sequences that work as multimers and get you a gene-specific DNA binding domain. So this DNA binding domain can be mutated and designed to target any part of the genome. We then fuse these with functional domains to give specific activity. And for TAL effector nucleases, we fuse them with the FOC1 endonuclease. TAL1s will act as, will use TALs as pairs, and therefore you will have a right hand and a left hand TAL, and in combination with their 2 FOC1 nucleases will cause a double-stranded break, which will leave 4 base pair 5' overhangs after cleaving. And TALEN pairs can be introduced into eukaryotic cells as either plasmids, or more recently, we've developed a means to make these as RNA molecules and provide those as the delivered material into cells. Now, we focus on TALENs, specifically for genome editing, as they have a number of advantages over the CRISPR system. One being is that there's no sequence restrictions. These DNA binding domains can be designed to any part of the genome. And because of that, TALENs allow us to get an increased proximity to our desired insertion sites. We'll talk more about that in some subsequent slides, where Cas9 will have to be designed to those protospacer adjacent motifs or be dependent on PAM sites. TALENs can be designed to cut at any part of the genome. And because of this, we do see improved homology-directed repair when using TALENs relative to CRISPR. Now, when we look at homology-directed repair, and we've got our CRISPR-Cas9 or our TALEN nuclease, and it's cleaved the genome, we need to be introducing a donor DNA at the same time that's going to have the appropriate left and right homology arms flanking our insertion site, which will allow for this new piece of DNA to be incorporated into the genome and effectively cause the knock-in effect that we're looking for, either to add a reporter gene, create a fusion protein, disrupt these functional domains, or change SNPs. And for this, there's a number of different types of donor templates that a scientist could choose to use. The first is single-stranded DNA. These are generally less than 200 nucleotides long, and this is due to the the synthesis limit that we see for commercially available custom DNA oligos. Those generally cap out at 100 nucleotides, and some vendors are able to get that up to 200 nucleotides long. These single-stranded DNA donors are used to cause small changes, such as SNP changes or amino acid changes, or small deletions in the genome. The second donor type will be double-stranded DNA. These are in the range of 200 to 5,000 base pairs, or 0.2 to 5 kb. They can be sourced either as linear plasmid fragments or generated by PCR. And these types of donors are chosen if you're trying to do a gene insertion, create some sort of fusion protein, or add reporter constructs to your gene of interest. Next, for even larger insertions, some scientists will choose plasmid DNA. These are generally used for knock-ins from the 1 to 10 kb range, although theoretically there is a higher limit, although the efficacy becomes very limited. Plasmids for donor DNA are generally produced by doing DNA synthesis, some sort of subcloning into a propagation vector, and then purification of that plasmid from host E. coli. And again, these are done to do larger gene insertions or add larger reporter constructs. And then the last would be a viral vector, and we're seeing more scientists using AAV viruses to accomplish their knock-ins. These are generally for insertions from 1 to 4.5 kb, and these are produced by doing DNA synthesis, subcloning into viral vectors, and going through a viral production process. And these are generally done for larger gene insertions. And these have gained advantage for some applications due to the fact that the virus can transduce the cells and therefore serve as their own delivery vehicle. For our presentation today, and most of the R&D work that's been done, that we've done, has been focusing on these single-stranded and double-stranded DNA donor types. Now, when we talk about donor design, what the questions that we get from a number of scientists that we go out and talk to is they want to know how they should be best designing this molecule. How close does my PAM site need to be to the insertion site? How long do my homology arms need to be? How well does my guide RNA need to cut? Should I be using single-stranded versus a double-stranded donor? And collectively, the scientists that we work with are asking, can you help me design my donor molecule? What does this donor molecule need to look like to be successful? So what we did as part of our R&D group is we went to establish consistent and simple rules for HDR donor design. We wanted to make sure that we had a simple and easy to follow rule set that was going to be consistent across a wide variety of edits, that we didn't have to rewrite the rule set for each different type of edit that someone may want to do. We took these rule sets and we developed a novel design tool that we put on thermofisher.com that has an intuitive user interface that makes it easy for users to design their own knock-in experiments. And then we made this tool free and available to all scientists so that we can share this information and make sure that it's applicable and accessible to everyone. So to look at this data and kind of to set up how we went about investigating this, we set up a number of different experiments to kind of screen different donor configurations. And this first experiment here is we generated a knock-in cell line that had a crippled GFP protein put into its genome. And what happened here is that we have the GFP will require a 6-base insertion to reproduce its fluorescence activity. So at this position 0 here, we're looking to do a 6-base insertion, and this will activate GFP, and the GFP protein will now fluoresce. Across this GFP gene, we looked to identify all of our guide RNA sites that are both 3' and 5' of this insertion. So as we look across the 70 base pair region, we've got guide RNAs at -34, -31, and all the way up to +21 and +30. And these are basically guide RNAs that are going to have cut sites that are that many bases away from our position 0 where we're trying to do the insertion. So we took all of these guide RNAs across this entire region and screened them for their cleavage efficiency. And we can see across the board that a majority of these guides have indel percentages greater than 60%. These guide RNAs are able to cut at these different sites across this GFP protein. Some of them perform less well than others, like this -19 and -14, perform less well than we see at the -31 or the -20. And we've got some particularly poor performing guides here at this +21. But overall, we kind of have a different array of guide RNA cleavage efficiencies. Now, when we look at our percent GFP positive cells, or the cells that have had successful and precise HDR occur, we see that the guides that were in closest proximity to our insertion site, so our guides that are -7, -3, +3, +5, and +8, are giving us the highest knock-in rate relative of this panel. If we look at some of the more distal guide RNAs, like our -31 or our +30, we do see some GFP repair there. We do see some HGR for those guides that are more distal, but it's significantly less than the guides that are going to be within that close proximity to our insertion site. Now, what's interesting about this is our +3 guide RNA, we do see a certain degree of knock-in here, but it's much less than any of the neighboring ones, and this is likely due to the low cleavage efficiency of that guide RNA. So, to kind of summarize these data, the rule sets that we've developed and the learnings we have from this is that we would recommend using an insertion site within 10 bases of the DNA break, or have your engineered nuclease cut within 10 bases of your intended insertion site, and to use the highest cleaving Cas9 guide RNA, or the highest cleaving nuclease available to cut this particular loci. And if you want to see more of these data, we have all of this information and a couple more experiments published in our Journal of Biotechnology article, which you can access via thermofisher.com/TrueDesign, which has a link out to the original paper, which is an open access and available for download. Now, we repeated this experiment, doing something a little bit different now with double-stranded donors to understand if these same rules apply. So looking across this segment here, we are trying to do a knock-in experiment where we're knocking in a donor cassette that has a resistance marker, a self-cleaving P2A, and then a GFP reporter into the N-terminus of the ACTB gene. So we're trying to knock this in immediately after our start codon. And we identified all of our guide RNAs across this loci and looked at the cleavage efficiency for all of them. And as you can see in this graph over here, we get about an average cleavage efficiency of about 60%, although guide +19 clearly does not cut, as we saw no cleavage with that particular guide RNA. And then when we look at our percent GFP positive cells, again, we see that we get the highest percent GFP positive cells when our cleavage site is close to our insertion site. So we see the highest knock-in rate with our +1, and we see a decent amount with the -5. There is some signal at the -20 or -27 guide RNA, but again, we're seeing over 50% GFP positive cells at that +1 cutting guide RNA. Now, what's interesting about this donor, as this does have a resistance marker for puromycin selection on it, is that we can apply puromycin selection to all of these cells, and we see that we can pull these populations up to being greater than 80% GFP positive. So even our cells that were cut with a -27 guide RNA and we're getting less than 1% knock-in, we can pull that population to being 80% GFP positive by applying this puromycin selection. And importantly, our -19 guide, which has 0 cleavage efficiency, basically this guide RNA does not work, also kind of serves as our negative control here, and we've got no signal either with or without puromycin. And we'll kind of touch on this system a little bit more in later slides. But again, kind of reaffirms our rule set here of have your DNA break within 10 bases of your intended insertion site. Now, if you're looking to model SNP changes, you are going to have a very precise location where you're going to want to have this genome editing occur. And this might be difficult to do with CRISPR-Cas9 if you're looking at something that's in a PAM desert. So what we have here in this table are a number of annotated SNP changes in the BRCA1 gene exon 11, which is known to be oncogenic and an important loci for a lot of oncology studies. And if we look at exon 11 here and try to identify the SNPs that we can edit with genome editing, we realize that with CRISPR-Cas9, we realize that there's a limited number that are actually accessible by CRISPR-Cas9 based knock-in. If we screen out part of this DNA for PAM sites that are within 10 bases of the SNP, we actually lose a large part of this genome because there's no PAM sites that are going to be available to work with the CRISPR-Cas9 system. For the ones that do remain, and we look for high efficiency cutting, or basically high cutting guides, we also eliminate another area of this region. And if we remove guides that have off targets, we've actually excluded a large part of this exon 11 that's going to be editable by Cas9. And again, this is due to that PAM desert, the low number of NGG sites that exist within this sequence. And overall, 70% of this region is inaccessible by CRISPR-Cas9 for successful knock-in editing. So what would you do if you're trying to model a SNP change that exists in a PAM desert? So looking at another model SNP that exists in a PAM desert, this PIK3R R348 missense mutation, nonsense mutation, excuse me, SNP change, we identified CRISPR-Cas9 cut sites that are going to be able to reproduce this specific SNP change. And we see that there's two guides that are available at the -17 and -20 position. And when we look at our precise HDR for this -17 guide, we see that we get less than 1% knock-in rate using CRISPR-Cas9 and that guide RNA to try to do that specific SNP. Now, for this particular exon in this particular region where we're trying to do this A to T conversion to cause this phenotypic, or excuse me, change the genome here, that -20 and -17 are the only two guides that are within this area that we could try to do this particular knock-in. But if we design these with TALENs, and we can design this TALEN pair to cut specifically at that SNP site, we're actually able to get an HDR, a precise HDR knock-in greater than 40% because we're able to target these TALENs specifically to that SNP site and encourage successful HDR. We also get a certain degree of HDR with error, where we're getting imperfect junctions, either the 5' or the 3' of that insertion. So basically we had HDR occur here, but we did not get the SNP change that we were intending to. And overall, we see the total HDR rate being around 50%. When we look at our indel frequency, so basically all the types of edits that occurred from having a double-stranded break but did not take up a donor and did not have HDR, we see that the indel rate's actually pretty high for our CRISPR Cas9 system. So this guide RNA is cutting. It is an effective guide RNA for causing cleavage at that site, but it doesn't effectively take up this donor molecule and get the successful HDR. And then the remaining fraction for both these experiments is going to be wild-type alleles that were unedited or remained unchanged after the CRISPR-Cas9 or TALEN-mediated editing. And again, something to keep in mind here is that this guide RNA is well-performing. We do see greater than 60% cleavage or indel formation for this guide RNA despite having no HDR. Now, when we try to normalize this a little bit, we are going to design TALENs and CRISPRs to cut the same site. We continue to see TALEN being more efficient in having precise HDR than CRISPR. So for this example, we looked at AAVS1 and identified guide RNAs that would be cleaving at this loci, and then designed TALEN pairs that would cut at the same site. So basically, we identified CRISPRs, and then we designed a TALEN pair that would be cutting at that same loci and then try to do a knock-in there. And for this, we see that our precise HDR is about 40% for TALEN and less than 20% for CRISPR. So we have a significant improvement in our HDR rate when we choose TALENs. Our HDR with error is pretty similar with both tools. And our indel rate kind of fills out the rest of this fraction where we have a higher fraction of indels occurring with CRISPR relative to TALENs. And again here, our guide RNA is working pretty well. Actually, both of these tools are cleaving at about 90% at this loci, but we get a higher fraction of precise HDR with TALENs than we do with CRISPR. And if we look at other loci as well, like IP3R2, and we do the same kind of design, we see that TALENs will outperform CRISPR in terms of the total precise, or the fraction of precise HDR in these cell populations after editing. If we look at a summary of these data comparing CRISPR and TALENs, we can see that kind of across the board here, looking at a number of different targets, we have some high-performing CRISPR-Cas9 designs. We're getting total editing greater than 90% for a number of these, but the HDR rate, either precise HDR or HDR with error, is less than 30% across the board, with some of these having very little to no HDR successfully occurring. But if we look at a similar panel of TALEN-based genome editing, we see that our precise HDR is greater than 20% almost across the board. We have some fraction of HDR with error, and our total indel frequency is significantly less. So overall, our TALENs are able to give us a higher rate of HDR and can be a more effective tool for knock-in editing. As these have proven to be a useful tool for knock-in editing, we've recently developed a TALEN mRNA production service to make ready-to-use TALEN mRNAs for genome editing. And again, here, some data summarized demonstrating the percent HDR improvement with TALENs versus CRISPR. We have now developed this service that's going to allow users to get access to this technology easier. And the way that this works is that we will take the sequence of the genome that you're looking to edit to. We will generate constructs to produce those TALENs that would cut at that specific loci. And then we will perform an in vitro transcription and RNA purification off of that plasmid construct that we make and provide ready-to-transfect TALEN mRNA for genome editing purposes. And if you're interested in learning more about this TAL mRNA production service, I encourage you to reach out to your local rep, or you can email our services lab directly at gemservices@thermofisher.com. Now, going back to donor design, kind of the last outstanding question that we really wanted to test here was to determine the minimum donor homology arm length that's needed. And if you look at more dogmatic approaches to genome editing in the literature, you're going to see recommendations for single-stranded DNA having at least 50 nucleotide homology arms on either side of your edit. But what we know from single-stranded DNA and how these are produced is that long oligos are going to have a lower synthesis efficacy or efficiency and become more expensive the longer that they get. And when you look at double-stranded DNA, that recommendation ends up being about 500 base pairs or more. And when you're trying to create homology arms that are going to be 500 base pairs or more, you're now talking about either going through a gene synthesis pathway and having it made synthetically, or you're talking about a multi-fragment cloning to piece all these together, as you're going to need a left homology arm and a right homology arm in your reporter sequence, all ligated together into one larger cloning complex. So what we decided to do is create a library of donors that are going to have increasingly smaller donor arms and test what's the minimum homology arm length that's needed for a successful knock-in. And so for this first experiment with single-stranded DNA, we created donors that had a flag EcoR1 insertion and then different homology arm lengths going from 45 down to 15 nucleotides long. And what we see here is that when we're using 15 nucleotide homology arms, we get very little knock-in rate. This percent knock-in is pretty low. When we go to 24%, we see that, or 24 nucleotides long, we see that improve to close to 20%. And when we go to 30 nucleotides or longer, we see that kind of plateauing out, where if you have at least 30 nucleotide of homology arms or greater, you kind of get the max knock-in rate that you're going to get with just kind of these donor design characteristics. When you look at double-stranded DNA, and you do a similar experiment, but we're working with a little bit longer arms, we see that at those short donor arms at 12 or 21 nucleotides, we get low knock-in rate. But when you get to 35 or higher, that again starts to plateau. We see a similar effect if you're using single-stranded or double-stranded DNA, where there seems to be a bit of a ceiling here that you reach, and longer homology arms don't necessarily add any benefit despite the increased level of effort that might be needed to produce longer homology arm molecules. So this leads to our last rule for effective donor design, which is to use homology arms that are at least 30 to 35 nucleotides long, as these are sufficient to get effective knock-ins. So with all of that, we now have our 3 simple rules for HDR donor design. 1, have your DNA break within 10 bases of your desired edit site, so choosing a nuclease that's going to be able to give you that cut less than 10 base pairs away from where you're trying to change the genome. 2, use the most efficient enzyme possible, whether that's CRISPR-Cas9 or TAL effector nucleases, to give you that high cleavage rate at your desired loci. And 3, include at minimum 30 nucleotide homology arms on either side of your desired change on your donor. And so with these 3 simple rules, we wanted to make this accessible and effortless for scientists to use these rules for designing various donors. And so what we did is we developed a new platform that we're calling our TrueDesign Genome Editor that's now available on thermofisher.com. And what we did here is we made a software platform that allows you to search for your gene. We've got the human genome loaded here that allow you to search up any transcript and modify it as you wish. Once you've searched for your transcripts and identified your gene, you can then go ahead and craft your edit and do specific changes to the DNA sequence and change specific amino acids. And then lastly, the tool will go ahead and complete the design. It'll take in the design criteria that you've given it, the insertions, deletions, tags you want to add, and then it'll go ahead and design the appropriate CRISPR-Cas9 guide RNAs or the TAL effector nucleases that are going to be needed to generate the cut at that site, as well as design the donor molecule that'll be needed to accomplish that knock-in in the lab. And with that, I'm going to do a quick walkthrough of our TrueDesign tool. Again, this is live on thermofisher.com right now. The first thing that you would do as a user is you will then search for your gene of interest. We currently have this loaded with the human genome, and we will be loading additional genomes into this tool throughout 2020. But once you search for your gene name, for example, here I've got the REL-A gene, you'll be displayed with all of the various transcripts for this particular gene. And once you select the transcript you're looking to edit, we will then display this gene in a topology format showing you the introns, the exons, and the untranslated regions for this gene. And then you'll be able to view the specific DNA sequence as well as the amino acids for this particular protein close up. And then you're able to point and click and edit the genome as needed for your specific knock-in. So what I have on the sequence viewer here is we are looking at the C terminus of the REL-A gene. I put my cursor between that final serine and the stop codon here. And then what I've told the tool I want to do is I want to insert this 24 nucleotide sequence into the REL-A gene before our stop codon to create a fusion tag here. And then once I click insert, the tool will refresh and it'll show this new sequence that I've inserted excuse me, at the C terminus of the REL-A gene here, and now I've added these 8 additional amino acids for the flag tag before the stop codon of REL-A. Once I've completed crafting the edit that I want to accomplish, the tool will go ahead and design various CRISPR sequences and TALEN sequences that can cause cleavage at this site, as well as design the donor molecule that would be needed to accomplish this particular edit. So what the tool is showing here is the various CRISPR-Cas9 guide RNA sequences and where they cut, as well as pairs of TALENs that can do equivalent editing at this site. Now, remember when we talked about for the donors, we have those homology arms of at least 30 nucleotides long. So what we have here is our donor molecule relative to the original DNA, with both our left and right minimal homology arms flanking that flag tag sequence that we've inserted. When you're done doing your design, all of the information, your guide RNA sequences, the TALEN sequences, the donor for the oligo donor sequence, sequencing primers are all available in a comprehensive download, and therefore kind of compiling all of the information that you would need to accomplish this edit in your lab. We're going to be continuing to develop this platform throughout 2020, including different edit types, as well as connecting this with our thermofisher.com cart system, making it easier to obtain all of these materials in one click. So, at the beginning of our presentation, we talked about the required materials for knock-in. The first was going to be a specific and efficient nuclease system. And with that, TrueDesign will recommend either our CRISPR-Cas9 products, our TrueCut-Cas9 synthetic guide RNAs, or TALEN pairs, depending on the type of edit you're wanting to do and the design restrictions that may apply to that specific part of the genome. It'll then give you the repair template with your desired change. Like we talked about, it's got our single-stranded DNA oligo here that would be recommended. It's displayed as double-stranded DNA to describe what it would look like. And we've got our left and right homology arms here. And then 3 methods to deliver in the cells. And the tool will recommend either lipofectamine CRISPR-Max if you've chosen to go down a CRISPR-Cas9 editing pathway, or lipofectamine messenger-Max if you're going to be using the TALEN mRNA pairs. And so what TrueDesign here is kind of piecing all of these materials together to give you a comprehensive solution for knock-in genome editing and give you all the tools that are needed to have a successful experiment. With that, we're going to pivot a little bit and talk about how we can drive this knock-in efficiency to be even higher using our TrueTag Donor DNA system. So, if you remember earlier in the talk, we talked about knocking in a GFP fusion into the N-terminus of the ACTB gene. And we scanned along all of the various guide RNAs across this exon 2 of ACTB. We looked at their cleavage efficiencies here. And then I showed you a graph where we showed that the highest knock-in rate was going to be at that -5 and +1 guide. But what I also showed you was that if you apply puromycin selection to the cells that are edited with this donor that has a resistance marker in here, we're able to pull all of these populations up to 80% GFP positive cells. And applying this selective pressure dramatically improves the percent edited cells, as we're selectively excluding cells that did not have successful knock-in here. So even cells that had a poor knock-in rate, had 1% knock-in, like our -27 guide RNA, we're able to pull that population up to be greater than 80% GFP positive cells by applying this selection. And so to kind of look at this a little bit further and how we can apply this and make this flexible, we tried to look at tagging genes at their N and C terminus. So looking at TrueDesign Genome Editor here and looking at the ACTB gene, we made designs for N-terminal knock-ins and C-terminal knock-ins. And what we were able to do is we're able to show that we could make GFP fusions at the N-terminus or the C-terminus of the ACTB gene and make GFP fusion proteins. So what we're looking at here is we've got U2OS cells that have been selected for with puromycin. They're about a couple days into the selective process, but you can see that we've got nice actin filaments forming in these U2OS cells that are visible because they're GFP fusion proteins. And this is we can achieve this at the N-terminus or the C-terminus of the ACTB gene. And when we look to quantify this, either with or without puromycin selection, we can see that when we apply puromycin selection to the N-terminus fusion, we're able to drive that population to greater than 80%. And when we do this at the C-terminus, we're able to drive this to being greater than 99% positive GFP cells. So the selective pressure here really allows us to get a purified, knocked-in cell population without going through any kind of clonal cell isolation process and having a really purified cell pool that's all knock-in positive cells. When we look at this as kind of like a time course, on our day 0, we have 0 GFP positive cells because they haven't been knocked in yet, but we're preparing our donor DNA, we're delivering it into the cells. If we look after day 3 or 4, we start to visualize and we're able to see GFP positive cells. The knock-in has occurred, the DNA has been repaired, this fusion protein is now being expressed. We now have a number of ACTB GFP molecules in the cells, and therefore we see GFP positive cells, and about 25% of these cells are GFP positive at this point. We start to apply our selective marker here, and we're therefore killing off cells that don't have that puromycin resistance cassette. And at day 10, we now have a stable cell pool that's greater than 99% GFP positive cells. So we're able to create a very pure cell population that are all knock-in positive. Taking this beyond the ACTB example, we also attempt to tag a number of different genes in different subcellular compartments, looking at proteins like laminin in the nucleus, looking at mitochondrial proteins like ATP5B, looking at cytosolic proteins like, in addition to ACTB, such as CCT3, and microtubules like looking at tubulin. And across the board, with or without puromycin selection, we're able to see these fusion proteins. We're able to successfully knock these in frame, getting proper junctions, and make effective fusion proteins using these TrueTag designs. We also took TrueTag and combined it with an RFP protein instead of just GFP. And so what we're able to do here is start to do a couple different like dual stain type experiments. So for this example, we've got a histone protein with GFP, and then we've got RFP on our actin loci as well. And then when we dual tag these in 293 cells, we can see our histone GFP protein in the nucleus. We've got the RFP actin in the cytoplasm. And when we do a merge of all of these, we can see appropriate co-localization, or excuse me, appropriate compartmentalization of the nuclear versus cytoplasmic signal of these various fusion proteins. So we can see here on our far right-hand side that merge where we've got red cytoplasmic and green nuclear signal, creating kind of interesting and unique cell models by knock-in genome editing. So TrueTag donors are generated by PCR. As we're using these 30 nucleotide homology arms, we're able to do this and make these donor cassettes specific for any gene of interest by PCR. So you'll have to take your forward primer and a reverse primer that are going to have consistent regions for the donor template, and then you customize the 5' end of those primers to be specific to your gene. And the TrueDesign platform actually has a tool for designing TrueTag primers. Once you go through the PCR and column cleanup, you then have a transfection-ready donor that's going to have your left and right homology arms added, flanking your insertion, which is the reporter P2A resistance marker construct. And this entire process for making this donor we've reduced to being less than 3 hours and includes no cloning steps. So, kind of in summary for this little bit, we have our new TrueTag donor DNA systems, and this allows you to get greater than 99% edited cells. We currently have solutions for GFP, RFP, and luciferase fusion proteins, and you can generate your donor molecule in less than 3 hours. Each of these kits includes 4 different donor templates, 2 for doing N-terminal tagging or 2 for doing C-terminal tagging with puromycin or blastocytin resistance proteins. And the kit also includes PCR master mix for producing your donor, again, doing that PCR to add the homology arms. Includes the cleanup module for cleaning up the DNA prior to transfection. And we include positive control primers for ACTB so that you can reproduce that ACTB example that I showed at the beginning here, where we're able to get greater than 50% knock-in at the N-terminus of ACTB using the TrueTag system. So the primers for that particular design are included with each system. So moving into our last bit here, we're going to talk about 2 case studies where we're looking at pluripotent stem cells and T cells and the TrueTag donor DNA system. So working with our stem cell colleagues who are looking to develop models for studying fate determination from pluripotent stem cells, we designed an experiment where we're going to take iPSCs. We would edit them with our TrueCut Cas9, a single guide RNA, and TrueTag donors, apply selection for 10 days. We then have a TrueTag PSC population, so pluripotent stem cells that have been edited to be TrueTag knocked in. We would differentiate these into neural stem cells and then ultimately terminally differentiate these into astrocytes. And so what we did for this astrocyte-specific experiment is we decided we were going to tag the GFAP protein, which is expressed in astrocytes, but not in neural stem cells and not in pluripotent stem cells. And for this, we chose to do something a little bit different and do a C-terminal knock-in. So what we did before the stop codon is we designed a new donor that would have, again, those minimal left and right homology arms, our reporter sequence, and then its own promoter for the resistance marker. And what this does is allows us to make a GFAP-GFP fusion, but since the resistance marker has an exogenous promoter, it can be selected for independently of GFAP expression, which means we can select for these knock-ins when they're in the pluripotent stem cell phase and not just at the differentiated phase. So what we did is we made our TrueTag iPSC cells, and we can see here under fluorescence and bright field that these are not GFP positive at this time. We went through our neural induction into NSCs, and then we went into our astrocyte differentiation. And after about 18 days of going through astrocyte differentiation, we start to see a GFP signal. We've got less than 1% cells being GFP positive at this time, but remember they're moving from being a neural stem cell into an astrocyte. So we're starting to see some of that GFP signal turn on. And when we get to about day 45 of differentiation, greater than 90% of our cells are GFP positive, showing that we successfully knocked in to something that's only being expressed in our astrocytes. The second thing we did was then to confirm if we'd successfully tagged GFAP and it wasn't just being turned on non-specifically in an astrocyte-like cell. So what we did is we looked for our GFAP EM-GFP signal. We then stained the cells with a GFAP antibody. And when we do our overlay on the far right-hand side, we see really nice co-localization of the EM-GFP signal and the GFAP antibody signal, showing that we're getting specific tagging and specific fusions made of the GFAP protein with our GFP donor. Again, demonstrating how we can do a knock-in at the pluripotent stem cell phase and then see the appropriate reporter signal at the differentiation point into astrocytes. For our second example, we're going to be talking about primary T cells, and it's been a really important, we've had a number of conversations with scientists about the importance of having complete solutions for T cell editing, especially when we talk about CAR T development and looking to use these use genome editing and immuno-oncology to make therapies. And so what we've done here for this general workflow is that we're able to isolate and activate T cells using Dynabead-based kits that are able to purify these cells from donor cell populations and activate the T cells. We can then do a T cell transfection using our TrueCut Cas9 and our guide RNAs, as well as the TrueTag Donor DNA system in the Neon. And then after 48 hours, we're able to assess the cleavage efficiency as well as any functional changes to the cells by either looking for our GFP reporters in the TrueTag system, as well as looking for cell surface proteins that we're editing. So for this example, what we did here, again, we isolated and activated Dynabeads. We then did our T cell transfection, and then we're analyzing them with the Attune NXT flow cytometer. And if we look at the left-hand side with our untransfected sample, we see that our T cell population that we're starting with is about 92% TCR positive. And if we go in with guide RNAs against the alpha or beta chain of the T cell receptor, we see that we're able to knock out that TCR signal by 92% or greater when we're targeting either the alpha or the beta chain of the T cell receptor gene. So if we look at this example in the middle graph here, we're seeing that there's about 2% cells remaining TCR positive, and when we look at the beta chain, we're seeing about 7% cells remaining T cell positive. But overall, using either of these guides, we're able to demonstrate a greater than 92% functional knockout of the T cell receptor with our workflow. When we look at donor-derived T cells and donor knock-in, we're able to knock in the TrueTag system, again, looking at this ACT-B model here, and we're able to knock this into the ACT-B locus of these T cells. And we see with our buffer R that's included with the neon system about a 5% GFP positive cells, and buffer C17, which was currently under development to improve our knock-in or improve our transfection systems and improve our knock-in, we see that actually increasing to about 29% GFP positive. Looking at a second locus, the RAB11A locus, it's a similar story, where using buffer R, we're able to get about 30% GFP positive cells, and with our buffer C17 that we're currently developing, we see about a 15% improvement in that as well, demonstrating kind of this model for developing new tools for genome editing in T cells as well as delivery of materials into T cells. So we talked about knockout and knock-in here, and so for this last experiment, we really tried to piece it all together. So if we look on the plot on our left-hand side here, our control, we've got 96% of our cells being TCR positive, and when we go ahead and do our knockout only, we get about a 93% TCR knockout. We do a combination of knockout and knock-in using the buffer R and a GFP donor. We see about a 94% TCR knockout and a 6% GFP knock-in. And when we do this with the buffer C17 that we're developing, we see about a 94% TCR knockout and close to 30% GFP positive cells. So again, we're able to demonstrate kind of the combinatorial nature of knockouts and knock-ins in T cells, really getting to the promise of engineered T cells for various therapeutic applications using these combination of editing tools. So, in summary, for this presentation, we reviewed knock-in genome editing. We talked about the basics of CRISPR-Cas9 as well as TAL effector nucleases. I then went through and presented the rules for successful HCR donor design and the R&D data that was used to generate this rule set. We talked about the TrueDesign tool and how you can use that today to design your own knock-in experiments to create SNP changes or amino acid changes or introduce a TrueTag into your system. Number 4, we showed you how to obtain up to 100% edited cells using TrueTag donors and applying selective pressure to pull those knocked-in cell populations up to 100% edited. And then lastly, we applied TrueTag donors to pluripotent stem cells and T cells, again demonstrating the versatility of these various knock-in methodologies. And with that, I thank you all for your attention, and I believe we will now open it up for the live Q&A.
Oh, thank you very much, Matt. That was great. We had a lot of questions come in, so we will get to those right away. If you still have a question you'd like to ask, you can do so. Just click on the Ask a Question box located on the far left of your screen. Okay, Matt, our first question came in kind of early in the presentation, so you might have already addressed it. But the question is, what's the maximum length of the insertion sequence?
Right, great question. So for the TrueTag system, we are working with donors with a maximum length of about 1.5 kb, and that's where we've done most of our development work around the double-stranded donors in that configuration. We're looking at that 1.5, maybe 1.6 kb in length. Our services team have done knock-ins up to 5 kb for custom projects for our different scientists, but the development work that we focused on here is going to be that 1.5 kb size or smaller. And then for the single-stranded donors that we talked a lot about too, the maximum insertion site that we've played with there is about 30 base pairs of insertion when using a single-stranded donor. So if you're using single-stranded DNA, we're looking at about 30 bases, and double-stranded donor, we're looking about a total of a 1.5 kb insertion.
Okay. Next question, do you observe differences in guide RNA efficiency for the same target depending on the cell type?
Depending on the cell type. So in general, there will be cell-to-cell differences in guide RNA cutting, but the trends generally remain the same. If you've got a well-performing guide in cell type A, it'll generally be a well-performing guide in cell type B, and the same will be true for poor-performing guides. If it's not cutting well in one cell line, it's probably not going to cut well in your other cell lines. The absolute cutting efficiency for a particular guide will vary from cell to cell. And that's generally dependent on the overall delivery efficiency, how well you can get that Cas9 and guide RNA complex into your cells, as well as small changes in kind of like chromatin conformation or configuration, and depending on the cell type. But the trends remain the same from cell line to cell line.
That makes sense. Okay, this next question, I believe, is looking to address the TALEN or the CRISPR approaches that you talked about for nuclease cleavage. It's asking which technology has a lower off-target rate.
So that's a design-specific question for CRISPRs or TALENs. Now, the benefit, kind of the overall recommendation is that TALENs will have more specificity, as you do require a pair of design nucleases to come together at a specific spot to cause a double-stranded break. Since you need that pairing, the right and the left TALEN, you are adding a certain degree of specificity there, which would reduce your overall off-target profile, because you would need that exact pairing of 22 nucleotides to be within 5 bases of each other in multiple parts of the genome to get off-target events to occur not, where you wanted them to, versus CRISPR, where you're just looking at that 20 nucleotide
the odds
sequence.
Of that sound pretty thin.
Yeah, exactly. And then yeah, and then so for CRISPR, obviously you're working with that 20 nucleotide sequence, so there is a higher likelihood of that occurring. And then also since it's a DNA-based pairing, you can get blebs or bubbles forming between the RNA-DNA duplex, and that can also lead to some off-target specificity. But it does again depend on the target and where you're looking to cause the cut to make like an absolute recommendation of an off-target profile.
Right. So next question, there's actually two that I'll kind of ask you at the same time. They both have to do with the TrueGuide design tool.
The TrueDesign tool, yeah.
Oh God, sorry, yeah, TrueDesign.
No worries.
Any truth. The TrueDesign tool, yes. The first part is, what if I want to design a knock-in experiment for something other than human? And then kind of along the same lines, do you plan to expand your knock-in software to non-coding regions?
Okay, gotcha. So yes, the current design tool, TrueDesign, is loaded with the human genome. We are still in development of that tool to build out additional features. So we are planning on adding additional genomes, specifically mouse, at least within 2020, and we're looking to add like rat, zebrafish, some other model organisms to TrueDesign platform over the next couple months. Into non-coding regions, that's something that we have talked about with the development team, and we're looking to understand what that would need to look like from a user UI experience to enable editing in non-coding regions. But it's something that's definitely been on our roadmap of what we'd like to take this tool.
So I can see where the question might be, how do you orient yourself without exon, intron boundaries, or kind of some of the land.
Exactly. And a lot of what we talked about when I showed in those screenshots is we're really trying to help users orient themselves in the genome so they can make a specific edit. And we've anchored that a lot around the exon intron boundaries. So if we are going into a non-coding region, we don't have that same kind of landmarks and orientations to put the user in. So we're trying to figure out kind of the best way we would solution for that. And then once we come up with that, we'll be able to support that kind of edit type.
Right. Okay, so this one has to do with the TrueTag kit. Can the resistance or selection insert, so the puromycin, be removed after initial knock-in? So I guess kind of getting a clean signal only without the resistance marker.
Yes. So there's two ways that a user could kind of solve for that. One, as the donor, so the donor templates that are provided in the kit obviously have the, your reporter and the puromycin selection or blastocytin selection marker. You can design those donors to omit that, as it is just a PCR. You can design your primer to kind of skip over that puromycin or blastocytin resistance marker and just amplify and add homology arms to just the GFP or just the RFP. So you could do your knock-in without the puromycin or blastocytin resistance cassette at all, if you so choose. Now, you won't be able to do any selection on that front since it won't exist in your cells. But if you were worried about that and just wanted to use the GFP part, you absolutely could do that. The
Okay.
second is that our C-terminal donors have LOX-P sites flanking the puromycin or blastocytin resistance. So if you're doing a C-terminal knock-in, you could do a second transfection with Cre recombinase and cut out that puromycin or blastocytin cassette using Cre LOX-P recombination to remove that from your, your purified cell or your cell line that you've made, and you no longer need that selective pressure to be applied. You could do that as well to remove the puromycin or blastocytin.
Okay. I'm going to try and squeeze in a couple more here.
All right.
For your knock-in, are you sequencing the results to check for errors in the junctions?
Yes. So when we did the development and we're looking at the TrueTag system as well as a lot of those SNP experiments that I showed you with TALENs, we were doing sequencing to get the fraction of, perfect HDR, HDR with air, indels. That was all sequencing data. And so for those SNPs, we were looking for basically the perfect repair of that SNP or those base insertions. And when we did our TrueTag product development, we also did sequencing of that to look for how well those junctions are occurring at the 5' or 3' end. And overall, the vast majority, we were seeing 85% or higher having perfect junctions, so them being integrated appropriately. And then also you have to keep in mind is that since we're making fusion proteins and we're running this all on one open reading frame, is that you have to get perfect junctions at the the 5' end there to get expression of the GFP and expression of the resistance cassette. If they're imperfect junctions that cause frame shifts, you won't see your GFP signal and you won't be able to do any kind of selection. So any cells that have imperfect junctions are going to be selected against.
Okay. I'm going to squeeze in one more question, but I'm sort of
All right.
two-part question.
Okay.
It came in when you were talking about the stem cell experiment.
Okay.
They asked if it's a stem cell specific promoter. If so, which one was used?
Oh, yes. Okay. So that donor that we showed for the iPSC and the astrocytes, so that's using an EF1 alpha short promoter. So it's
right.
It's not a stem cell specific promoter. It's a promoter that should be expressed in a wide majority of cell types. And the idea is we want that promoter to be on when the cells are still not differentiated, but for us to be able to continue to apply selective pressure or choose to apply selective pressure at any of our differentiation stages. So we chose something that should be at least somewhat ubiquitous to a lot of cell types to make that design applicable to different experiments.
Right. And then the second question around that experiment was whether the donor that you discussed is available in the current kit.
Oh, yes. So that donor design is kind of part of the second phase of our development. It's not currently available in the kits that we talked about. We're still building that out and doing additional validations on those particular designs, but that's something that we're looking to make more widely available in the future. If you are interested in it today, we can support that through our custom services. And so that can be a conversation with your local rep, or you can contact us at the GEM services at thermofisher.com address if you're interested in getting those materials.
Okay, excellent. Well, we are at the top of the hour. So before we wrap up completely, I want to thank you one more time, Matt, for the great presentation, and then, of course, thank our audience for joining us today. As I said in the opening, questions that we didn't have time to answer live, I noticed that a lot came in just in the last few minutes of the presentation.
A lot there, yeah.
We will be addressing these by email with the address that you provide at the time of registration. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Many of you have also asked about future viewing of this. It will be viewable on demand. So LabRoots will alert you by email when it's available for replay. So we'd encourage you, obviously, to watch it again or share it. Share the email with your colleagues who may have missed today's live event. And with that, we'll wrap up. I thank you again for joining and hope everyone has a great day.